piece, sort of, you might think of it as plumbing, but it turned out to be essential, a common coordinate system, a way that every time we record from a neuron, we image the neuron, we know exactly where it is. So we study in detail the connectivity of the mouse. We study in detail the electrical behavior of individual neurons. I'll show you that. We study the genes that they express, so-called transcriptional analysis, very powerful. We build brain observatories, where I'll show you what that is, where the mouse does some behavior. While it does its behavior, while it's taking some action, deciding whether to run past something or not, we can look at its brain. So we can infer its mind at the time that the animal has to make a decision. And then we're doing the detailed electromicroscopic reconstruction of all of that using powerful connectomic techniques. As I said, underlying all of that in the mouse, we don't have this with this precision in human. It's what's called a CCF, a common coordinate framework. We developed this over the last 15 years. It's in the 3rd generation now. It's pretty much standard now. In fact, NIH in its latest grants has just sort of mandated it, that everybody uses it. That has a big advantage. So you can see here, here we've superimposed 1,500 mice, and you can beautifully see the structures. This is, of course, one advantage you don't have in people, right? So these are all genetically, these are all transgenic. They have all the same genes, right? So it's like having twins, of having 1,675 twins of you, and I now superimpose all the brains, right? So, of course, humans don't have that. Humans, we have sex with whoever we or our ancestor wanted to, and so we have a huge diversity. That's one advantage of working in standardized mice. And so this atlas here has 500 million pixels. We can pinpoint things with this 10 micrometer accuracy, and there's a detailed ontology of all the different parts of the brain, roughly 800, similar number that it is in the human brain. Of course, the individual parts are smaller in the mouse brain. So then we can also do functional brain imaging similar to an fMRI, but with much higher precision. And we do that with every single mouse that we use. So just like some of you may have seen a map, for example, from the human visual brain. I've done it for my own brain, where you can pinpoint the different visual areas, V1, V2, V3. In the mouse, you can do very similar, except here you have everything is smaller. So this is a couple of square millimeters. There are 12 visual areas, in us there are 36. So it's not. I mean, its dominant sense, of course, is touch. It has whisker system, but it has a perfectly usable visual system that it does use. It's not quite as sophisticated as ours, partly because it doesn't have a fovea. Its resolution is roughly the same. It doesn't fall off dramatically as it does in us or in other primates. And then we have this detailed connectivity atlas where we have we've done this now in 3,000 mice, where we use the power of transgenics to inject in a particular part of the brain within the CCF. We inject a virus into a particular group of cells, either in wild type, where all the neurons are affected, or we use transgenic tools so that this virus is only expressed in a particular subtype of cells, let's say only layer 5A neurons, and then for 2 weeks this virus travels along the axons and it's fluorescent, so we can see it. Then we sacrifice the animal and we do tomographic reconstruction of the entire brain, 140 slices, takes us 1 day, and put that into a standard coordinate framework. And then we can say, well, if you inject in this particular part of the brain, let's say the area AM, which is the higher order visual areas, then here you can see on the same side, on the opposite side of the brain, where the connection matrix, if you want, this is the amplitude of the fluorescent signal in log unit. As I mentioned before, this is one of many things that's log normal distributed. So you can see cortex projects very strongly to cortex, projects on the same side, somewhat weaker to cortex on the other side, and systematically projects to various other areas. But here we're now getting it to the level of small groups of neurons. So this is just for visual cortex. Here we pulled it out, all the inputs into primary visual cortex. In your case, it's at the back, just above your bump. In the human brain, all the outputs. So typically for any cortical area, lots and lots and lots of different inputs. As a whole, lots and lots of different outputs. Even the eyes This is true for us as well. The retinal output doesn't just go to your visual cortex, that's one of 20 structures. There are lots of other structures that the optic nerve, the output of the eye, projects to. This is probably too bright. Because what we can do now, we have this fantastic technology. This is 3 individual neurons. It's very difficult to appreciate here with the light. Here you can see the dendritic trees. They're in a part of the brain called the claustrum that Francis Crick and I were very interested in. These neurons project throughout the entire cortex. So these are individual neurons where we can now, using powerful techniques where we cut up a brain into 14,000 slices, and can reconstruct it. And so we know where the entire output projects. Because traditionally what happens if you do neuroanatomy, you take a piece of brain like a slice, and you stain it and you reconstruct the axon in that little piece of slice, not the whole brain, because that's very, very, very expensive. Today, with appropriate computer technology and others, we can reconstruct, and we see this is called the crown of thorns neurons, because they project incredibly widely. This just makes modeling, once again, specific but very widely, makes it challenging to model. But we need that sort of data, so we're doing that at a whole scale. So then what we're doing in both mouse, and we're also doing this in humans, I'll talk a little bit more about that in a second, where we systematically do take slices of brain from always the same place, like visual cortex, a particular part of visual cortex, or the input to visual cortex, LGN, or the output, and under standard condition, we put it in a dish and we record from it using a standard electrical protocol, and we inject it with dye so we can reconstruct the 3D, the local dendritic tree, the local dendritic arbor. And then we also want to suck out the genes that are expressed, the mRNA inside the nucleus or inside the cell, thank you very much, and to genetically characterize it, because biologists believe that ultimately the best way to understand the different types of neurons is to look for the genes that are expressed and characterize them using that, which is a high-dimensional space. Typically 10,000 genes we can see using our deep measuring tools. We can see 8,000 to 10,000 genes being expressed in each cell. So you have a classification in this 10,000 dimensional space. Then we do electrical models of these neurons, because we want to use all these models to model the network. And then finally, all of this ends up, this is transcriptional, in a taxonomy, because ultimately we want to do data-driven taxonomy to be able to say, well, in this area there are 50 cell types, and over here there are 60 cell types, and this is how they relate. Some are different and some are identical. To try to understand the picture. For instance, in chemistry, we've done this, called the periodic table. We know everything here on the planet falls into one of 92 buckets. So what about biology in terms of cells? So you can go to our website here, and you can see something like this if you go to under this part of the website, cell type database. This is the back part of the visual brain of the mouse. Each color here is a particular neuron that's classified using some features you can select. Over here you can see thumbprints of it. You can see the electrical behavior in response to current injections. You can see the individual action potential. You can see some of its morphology. You can click on any one here and you can go deeper. So here you can see a particular neuron. You can get a picture of it, the 3D picture of it. You can get the FI curve. You can get the individual traces from all the data, how the cell responds to ramp, a step, white noise, pink noise, and you can download associated models using a standard SDK that we release with this. And then we do automatic fitting both for various types of single compartment models, like GLIF models, with refractor period, without refractor period, with adaptation, with spike adaptation, etc., a whole family of them. And we also do modeling of very detailed neurons, biophysical neurons. So 35 years ago, I did my PhD on modeling 5 of them, and today, using passive and active dendrites, and here you can see the black is the data, red is the model. These are the reconstructed for this cell. This is, these are the reconstructed morphology. Blue is apical dendrite, red is basal. Sorry, the other way around. So while it took me a PhD 3 years to do it, so now you can do this in a pipeline fairly automatically in hundreds of cells. And so the advantage is you get really high quality data. With internal quality control, typically we'll throw 50–60% of the data out that doesn't conform to our quality assurance and quality control criteria. All right. Can you advance? Seems to be up. I want to go back. All right. So we can, yeah. So this is just using data-driven, using various machine learning tools. So traditionally, people have done this by eye. Human anatomists have said, well, by eye, this is a pyramidal cell. That's a thick-tufted, that's a thin-tufted pyramidal cell. We're using objective criteria, data-driven morphological clustering. So yes, you cluster on the morphology, cluster on the electrical behavior to try to see what's the relationship between the two. So we can also do this routinely now in human, which is really pretty cool. So once every week or every 2 weeks, we get a piece of human brain. So the piece of human brain may be as big as the entire mouse brain, or it can be smaller like my pinky, or it can be sometimes big like this. So this is brain surgical tissue that's removed because the neurosurgeon has to access an underlying tumor or an epileptic foci, let's say in the hippocampus or amygdala. And in order to get there, he has to tunnel through the overlying cortex called the middle temporal gyrus. And so typically this piece of brain, typically gets incinerated as medical waste. We have a special procedure after lots of HIPAA and all sorts of other regulatory burden, but we get this tissue then within 20 minutes, and then we can record from it just like we can in a mouse, except it's much more robust. We can record from it from 2 or 3 days. We can see beautiful action potentials in interneurons, or here's an interneuron, a pyramidal cell. We can see synaptic connections that we can pulse. We can now use, we're developing fast-acting viral techniques, because of course we don't have transgenic technologies for human for obvious reasons. We have them in mice. So we need to develop virus techniques. For example, herpes simplex virus that only infects, let's say, the interneurons, or only a subtype of interneurons with a particular fluorescent dye. So then I take this slice, I infect it with this HSV virus, and then a day later only a particular subtype of neurons now glows fluorescent, and then I can target and identify them. So that's a rapidly evolving technology. And yeah, so you can reconstruct and model the human neurons just like you can do the mouse. This data will be first made available in October of this year to the general public. So as I said, we do this detailed transcriptional clustering. It's all the rage now in neuroscience, because we have these relatively inexpensive techniques that allow us to sequence tens of thousands of genes in any individual cell. So not just in bulk tissue like was done using microarray chips, but now in individual neurons or individual cells in the entire body. Some people are getting ready to do this in the entire body. We're getting ready to do it in the entire brain. Here we've done it in 2 brain areas, a primary visual cortex and a motor area. And I'm not going to go into the details. So here you have this the current result of our current clustering using 20,000 cells. As I said, operating in these 10,000 dimensional spaces roughly. So here, if you do a split, first you get non-neurons that separate out from neurons, and then if you go down the neuron pathway, you get the excitatory cells here that separate out from the inhibitory cells, and then there are all these different subtypes. The most interesting point for you is the following, that interneurons seem to be, so if you look at the key here, red means that that type is found in both cortical regions that are quite a way away in terms of cortical real estate. So it seems to be that interneurons are the same type, transcriptional defined, whether I'm in this part of the brain or this part of the brain, while excitatory cells differ, whether they're here and here. That makes sense because you probably also know that the output neurons of cortex tend to be almost entirely excitatory. And so the output cell from primary visual cortex, they go, let's say, to an oculomotor neuron, to the colliculus, its visual system. They have to carry a different zip code, and so they express different genes than the output of the motor cortex that goes to other nuclei. It makes it difficult to understand the effect of various gene deletion, and of course that's the challenge with human diseases, like all mental diseases. Almost all of them, you have hundreds of different genes. Each gene, if you have it, confers a very low probability of getting that disease, schizophrenia or ADHD or whatever it is. And it may well be because each of those diseases affects one particular subtype. There's some evidence to suggest that it affects only this subtype of somatostatin interneurons. And so you really have to understand that and hone into that in order to try to fix it. And so the challenge we're now facing, we as a field and we as an institute, we're the only one doing this so far because it's very expensive in terms of demand. We're trying to do all three at the same time to record the electrical behavior of neurons. This is mouse. We're doing this, trying to do the same thing in human. This is mouse, V1 and LGN. The electrical behavior, its shape, the morphology, and its transcriptional analysis, all in the same cell. In other words, we try to record from the cell, tickle it with electric current so we can see how it responds, gently put in some tracer so we can reconstruct it, and then literally, we suck out the cytoplasmic content. For humans, we have to include the nucleus. For mouse, it's not necessary. We take the cytoplasmic content to analyze it to get the transcriptome at the same time, because then we can combine all three modalities and do high-dimensional clustering. All right, so the other part we are doing, we're building this brain observatory. So we're very familiar with observatories, either in astronomy or in particle physics. They're children of the 20th century. Ultimately, they were created because it was too expensive and too big for individual labs to have these. Now, of course, you see one extreme is the LHC in Geneva that employs several thousand people with a yearly budget of billions of dollars. Now, we want to do something more modest, in order of magnitude more modest, but doing something similar in biology. We want to record cellular level activity, so not fMRI, but large fraction of individual neurons in a very routine, reliable, and high throughput manner, right? So that the experiment is reproducible. Because one of the crises of science, in particular biological and medical science, is of course the crisis of reproducibility, right? 2 out of 3 experiments cannot be reproduced. Partly that is because the data is never made available, and the data isn't generated under high throughput, standardized operating conditions. So we are trying to change that. And we're trying to do that in behaving animals, never been done before, and using 3 different sets of instruments. This is a technology, 2-photon calcium imaging, where we can record where, thanks to the magic of transgenic, the electrical activity of neurons is converted into flashes of green light that we can then pick up with 2-photon imaging. At the cellular level, at slow resolution, 30 hertz, but genetically identifiable. So we know now we're recording from a layer 2-3 neuron that expresses one particular gene, ENX1 gene, which means I can ship my mouse to another lab and they can do the same experiment and record from the same population of neurons. Or you do wide field imaging, single trial, I'll show you that. Or you do very high density. We build these very high dense electrical wiring that are thinner than a human hair, where you can record from 900 electrodes. We get things at resolution of 40 kilohertz compared to 30 hertz, and you get it all along a vertical column, but it's not genetically identified. That's the drawback of it. So this is basically the technique we use. It's a mouse that runs on a disk, 1 degree of freedom, and it's trained that if it stops and various pictures of me, then it gets water, and it can disregard everybody else. This wasn't at my insistence. I did this for fun, but mice can actually do this. So now, we have a team of 6 mouse surgeons that operate transparent skull into the skull, transparent window into the skull. So we can now swing a 2-photon microscope over there, or we can do these electrical recordings. The brain itself, of course, has no pain receptors. All right, we use a standard here that we have developed with the field. Is very important because if there isn't a standard, it's very difficult to read the data. So anybody who cares to experiment, you can just go to this website, Neurodata Without Border. It's an easy standard for data and metadata. So whether you're doing eFIS or OFIS or any type of recording, it's easily interchangeable. All right, so I'm not going to belabor the detail. This is an operation. It takes roughly 100 people, this pipeline. We we have to prepare these mice. So we take different mice, wild type or different transgenic animals. We do surgery, then they have to recover. Then we do the intrinsic imaging, so we know exactly where V1 is in the other higher order visual areas. Then we have to get the mouse used to running into the setup so that they don't freak out. Then we actually have to do the actual imaging. So here we also have to train it, whatever procedure we'd like to use. It has to respond to run past certain images or not run past certain images. Then we do the actual imaging, we do the actual cellular imaging, then we do the whole brain imaging, and then finally we sacrifice the animal to get the high resolution anatomy of that particular animal. So we have, we want this thing to be highly reproducible. We wanted to be able to go back session after session to the same neuron. So it requires a lot of mechanical engineering and gimbal and other fancy devices. So we can always go back with a precision of a few micrometer to the exact same neuron over multiple days. So I'm not going to belabor that. So here you can see very reliable. So here these are 8 different animals coming from 4 different transgenic lines. And here you can just see the pictures of them. And here you can see in each case we've identified the different visual areas. You can see it's remarkable, much more similar across animals than if I would do the same thing in humans. Partly because, of course, these are all transgenic animals. So it's one of the advantages. We have a standard protocol where we showed sort of the things that visual psychologists have been showing to people and to animals since roughly 100 years: grading, drift grading, static grading, sparse noise, dense noise, spontaneous, but also natural scenes. These are from the Berkeley segmentation data set, as well as movies, touch of evil. And this is intermixed over various days. So we can come here, you can see one of these. So this is, you can go to the website and download all this data using NWB and SDK. So you can see the mouse running on a wheel, they love to run. So here's the movie, the Orson Welles Touch of Evil running in the background. Here you can see individual neurons. So the flickering of the light here, that means that neuron just spiked. And so this, it's taken at 30 hertz. And here, of course, we track the pupil and where it looks. The pupil is proportional to how aroused the brain is, just like in us. That's why poker players wear shades. You don't want to give that away. And also we need to know where the animal's looking to precisely locate the visual stimulus. And so you can go, again, this is the back view of the visual brain, V1.